is very, very special. It's just like math, okay? I would say that you need to have a lot of practice. Okay, the more you practice, the more confident you will be. Okay, if you are too lazy to practice, you copy your friends, you're gonna screw up this topic altogether. Alright, so I recommend that you practice instead of copying your friends. Okay, and another tip is that in this topic, all the clues are given in the question, so you gotta read the question really carefully. Okay, so anyway, let's begin. Now, in this topic, there's a lot of different types of genetic problems. It's like an investigative sort of topic. So, in order to approach this topic, okay, there are some questions that you should ask yourself. Okay, can you capture? As long as it's captured, it's okay. Can you capture?
No.
Can I just leave it?
I don't know.
Can you capture?
Yes, now I can.
Okay.
Cannot, where I put that cannot.
Cannot, is
Cannot. Okay, yes. I don't
it?
know.
Should I stand here then?
So they no need to hear, they need to hear your voice.
Sell this table.
Okay, because our fun in Vietnam will be always quite long. Then, can you capture or not?
Can. Not.
Okay, just try your best, okay? Alright, can I move on? Okay, so these are the questions that I think are very good questions to ask yourself. So you have learned about this term called characteristic. What is that? Characteristic is something like your trait, your phenotype, okay? So in this topic, you have seen several traits, okay? So how many characteristics are being investigated? Okay, so usually in this topic, either you have one trait or you have two traits. Characteristic or trait. Okay, example, okay, height of plant is one trait. Okay, so that is characteristic. The second question that you should ask yourself is, how many genes are involved? Okay, it could be one gene controlling one characteristic. Now, what is that type of inheritance called? Very good, that is called monohybrid inheritance. Alright. Okay, please write all of these down now. This is the summary for this topic. Okay? Now it could be two genes controlling two characteristics, so that will be dihybrid inheritance. It could also be two genes controlling one characteristic, that will be epistasis, very good. So it could be epistasis. Okay, or you can call it gene interaction. Now, there's one that is not here, okay, but you will learn it for the next lecture. Right, that's the last part of the lecture note. It could also be many genes controlling one characteristic. Okay, so this one here, we will call it polygenic inheritance. Yeah. Okay, so this last one you will learn in the last lecture for inheritance. Okay? Now, the next question to ask yourself, right, because there are many, many different types of problems. For each gene, how many alleles are involved? Okay, so most of the time, how many alleles do you see in each gene? Most of the time, 2. Right, so most of the time, usually, you have 2 alleles. Alright, and usually these alleles have a relationship that we say they are complete dominance. Okay, they show complete dominance. Okay, so what does complete dominance mean? Okay, we'll talk about the relationship of alleles in question number 4. But besides 2 alleles, are there any other situations that we see more than 2 alleles? Yes, example? Yes, very good. So there could be more than 2 alleles also, so we call it multiple alleles. Multiple alleles. For example, IA, IO, and IB, basically controlling your blood type. Okay, so that is a number of alleles. Either it's a multiple alleles question or it is usually your 2 alleles. Okay, now the next question number 4, the relationship between alleles. It could be a pair of alleles that show dominance. One is dominant, one is recessive. So in this situation, we call it complete dominance. Right? So that's the first type of allele relationship, complete dominance. The second type, we call it co-dominant. Alright, and of course there is the last type that is not here, no space. It could also be or incomplete dominance. Okay, so the alleles can be complete dominance relationship. So there is one dominant, one recessive. Right? There could be co-dominant relationship, it could be incomplete dominance. So we'll look at each of these later on in the tutorial. Alright, now the next question to ask yourself is where are the genes located, because it does affect the question. So if it is located on the autosomes, what is an autosome? Non-sex chromosome. So in humans, your autosome is chromosome number 1 to 22. Right, that's the chromosome pair number. Whereas your sex chromosome in human is your X chromosome and your Y chromosome. That means the 23rd pair in humans. Okay, so where the genes are located is going to affect also. Okay, especially for sex linkage questions. Alright, so that's important. Now another thing to take note of is are the genes, let's say if you're observing more than one gene, are the genes linked? Now what does linked mean? It means that the genes, 2 genes or more, are located on the same chromosome. So if they are located on the same chromosome, it kind of affects how these genes are being inherited as well. Okay, they tend to be inherited together if they are linked. Alright, versus when they are unlinked, that means they are on different chromosomes. Okay, so we'll be looking at all these examples. And now these are very important ratios. Can you put a star? Okay, you're gonna see these ratios. Okay, so these ratios, right, the more you do, the more you will remember. Okay, so please just memorize. Now, if we are looking at a monohybrid inheritance, right, monohybrid means I'm only looking at one gene, right? For one gene, if let's say I have 2 parents that are heterozygous, that are crossed together in that one gene locus, the ratio of offspring that I'm going to expect is 3:1. Okay, it is forever 3:1 as long as they are on different as long as they follow a Mendelian inheritance pattern. Okay, I'll talk more about Mendelian's ratio later. So over here, if that monohybrid inheritance question, if I cross the parent that is heterozygous with homo-recessive, right, I'm going to expect an offspring ratio of 1:1. Okay, so you have to memorize this. Okay, if you memorize, you can do the problems more quickly. Now, if you're looking at dihybrid inheritance, which means I'm looking at 2 different genes, okay? Now, if I cross 2 heterozygote parents together, the offspring ratio that I expect will be 9:3:1. Okay, so please memorize this. And if I cross a heterozygote with a homozygous recessive, right, I'm going to expect a 1:1:1:1. So please memorize all of this. Okay, so let me see. Okay, now the next thing is this. What does it mean when I say it is a Mendelian inheritance pattern? By the way, you know who is Mendel, right? It's mentioned in your lecture. Who is Mendel? The plant guy. He's a monk, right? Remember Gregor Mendel? I think he's called the father of genetics. He's a monk. So he worked on pea plants and he discovered, the inheritance laws. So there are 2 laws that's mentioned in your lecture notes. Anyone remember them? Law of segregation, okay, and law of independent assortment. Okay, so these are the 2 laws. Now, what does law of segregation mean? Okay, something like that. Okay, it means that for each gene, okay, each gamete receives one of a pair of alleles. So for example, if you're looking at monohybrid inheritance, the parent is like this, right? When you're drawing the gamete, you have to separate the allelic pair. You get what I mean? Even if you're looking at dihybrid inheritance, A, B, B, right? for every allelic pair, okay, you're gonna have to separate it in each gamete. So you're gonna get this, 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 and this. You get what I mean? So you only get one allele of every allelic pair of a gene. Right? How about law of independent assortment? What does that mean?
It's random.
Random. So in case you cannot remember your law of independent assortment, we have studied it before. Something like this, right? So if you have two genes, okay, let's say for example, one gene here. Okay, let's say this is gene locus A. This is gene locus B. Can you see that they are unlinked? These two genes are on different chromosomes, right? So gene A is on the long chromosome, gene B is on the shorter chromosome. Let's say chromosome number 2 and chromosome number, 5, something like that. So if they are unlinked and you have independent assortment during meiosis, especially during metaphase 1, right? It means that, okay, the chromosomes can arrange in different ways on the metaphase plate. Right? It could arrange in this way where the paternal one, both of them are on the left, or it could be another arrangement where one paternal one is on the left, one paternal one is on the right. You get what I mean? So eventually, the gametes that you get will be all of these combinations. Okay, so that's the law of independent assortment. Alright, so this is the situation when you, so the moment you have Mendelian inheritance, you're going to expect all of these ratios. Now, which situations do you not get this sort of ratios? That means, in what situations do you have non-Mendelian inheritance? Linked genes, very good. If the genes are linked, okay, I'm not going to expect all of these ratios, okay, especially for the dihybrid ones. Any other examples of non-Mendelian inheritance? Epistasis is very good. Can you see that if we look at the table, okay, I mean, step number 7, all these are non-Mendelian ratios, alright? Basically, in epistasis, what you're going to cross, right, is two double heterozygote. That means, basically, for all of these, what you're crossing is a heterozygote parent with another. Okay, double heterozygote at each gene locus, basically. Right? What you're going to expect if it's Mendelian ratio, if you cross two heterozygote parents together, if it is a Mendelian ratio, what are you going to expect? You're going to expect 9331, right? But in epistasis, you don't get 9331. Instead, you get modified ratios. So these are your modified 9331 ratios. Why are they modified? It's because the genes interact in a certain way. They are either inhibiting or whatever. Okay, so because of that, you get modified ratio even though you cross two heterozygotes together. Okay, any other examples of situations where you don't get these Mendelian ratios? Last one, anyone? Sex linkage, good. Sex linkage, you don't get such ratios, right? Because it determines on whether you are XY or XX, okay? Whether you get the disease or not. Can or not? Did I cover everything? Oh, something else also. Last one. Last one. Any other situations you get a non-Mendelian ratio? Crossing over? No, nothing to do with crossing over. Okay. Over here, actually, the clue is question 4. Question 4, right? If you have co-dominance or you have incomplete dominance, you're not going to get a Mendelian ratio also. Example, if you cross these two parents together, you're going to get offspring ratio 1:2:1. Okay, I mean, we'll talk about more later. Instead of you're not going to get 3:1. Example. Okay, so co-dominance, incomplete dominance are also non-Mendelian inheritance patterns. You get what I mean? So how do I get Mendelian ratio? Okay, if you're looking at more than one gene, it has to be on different chromosomes. Okay, and it has to be not on the sex chromosome, basically. Okay, so anyway, did I cover everything? Let me see. Yes, I think that's all for now. Of course, what other parts are there in this tutorial that we will cover? We will be looking at pedigree also. Pedigree analysis. Now, what is pedigree analysis? Is that chart with the boxes, right? You know, the square and the circle. So this is an example of a pedigree. This one, pedigree analysis. Okay, so that will be that. By the way, I highly recommend that you complete the supplementary questions. You know, at the tutorial, right? In class, we'll be going through the questions in front, but additional supplementary questions at the back, I recommend you do. Actually, it's really not a lot. Okay, so please go and try everything. Okay, and what else are we going to cover besides pedigree analysis? We are going to do chi-square test, which is what you're going to do, what you covered in lecture today. And yeah, that's all. Ken? Any questions? Can I capture the screen? Alright, so I'm going to move on, okay? Now, first of all, can you look at the LO? Okay, can you actually put a star next to LO number A? Okay, in case you have not realized, right, Cambridge may ask you for definitions, especially in this topic. Okay, so some of the common words that they ask you for definitions is stated here. So make sure you memorize the definition if you are unable to explain your own words. Okay, so an example, these are your definition questions. So let's take a look at the definitions first. How do you define the term gene? Okay, let's divide the marks. Okay, gene, allele maybe 2 marks, gene locus maybe 2 marks. Basically, you can gauge from the number of lines, right? Number of lines. Now, how do you define gene? What is gene actually? Discrete information. Okay. Okay, specific sequence of nucleotide. Okay, yeah, let me just show you. Okay, so what you're gonna get, okay, just an understanding of it is actually on DNA. It is a specific sequence of nucleotides. The moment you change a single base, you're going to change, it's called a gene mutation already. Alright, and a gene has to be coding for either a RNA or it's coding for a polypeptide. Okay, now that's genes. Okay, now then how do you define alleles? Okay, a gene is a specific sequence of nucleotides, so perhaps this could be a gene, right? Then how do you define alleles? Alternative form of a gene. Okay, so this one is what you can at least write. Alright, so example, let's say I have a gene A that is responsible for hair color. Right? At this gene locus, right, the alleles that I can have is either capital letter A or small letter a. So that is what I mean by alleles. Okay? Okay, so your alleles are alternative form of a gene. It could be dominant allele, or it could be recessive allele, or it could be co-dominant, incomplete dominance, whatever. Okay? Now how do you elaborate? How can I elaborate this? By the way, allele big A and allele small a, do you expect them to have same nucleotide sequence? Identical? No. It's going to have a little bit of difference in their sequence. Okay? Same but different basically. So, yes, how are you going to elaborate about alleles? What else are you going to say about alleles? Exactly. It occupies the same gene locus on a pair of homologous chromosomes. So this homologous chromosome, let's say this is the paternal one, and this is the maternal one, so I represent different colors. Okay, so can you see that this gene locus is exactly where the alleles will be found? How else can you elaborate this one? I don't think anyone will write, but you can write about this. So basically, in a diploid individual, you can have a maximum of 2 alleles. Can you have 3? You can have 3 types of alleles present in the population, but only 2 can occupy the chromosome. Get what I mean? So you can have many, many different alleles, but only 2 can be found in the chromosome of a diploid individual. So for organisms that are haploid, how many alleles can you find? I repeat, if let's say there is an organism that is haploid, how many alleles can I find in a haploid organism? 1. Okay, so it depends on the ploidy of the individual. Last one, this one the easiest of all. How do you define gene locus? Position, yes. So it is the position occupied by a gene on the chromosome. So basically the alleles will occupy the same relative position. Okay, Ken? Yeah. You can see this board.
I'll zoom in.
Okay? Okay, last 1 minute. Okay, so now I'm just gonna recap this, okay? Now this is 2 ways at which I can represent homologous chromosomes. Sometimes you see it like this, okay, on top like this. Sometimes you see it at the bottom like this. What is the difference, Okay, so the bottom one shows you homologous chromosome after replication has occurred. Okay, now if you have this image here, what should I label for my alleles? Let's say these and these are exactly the same chromosomes, just that this is the replicated format. Over here, what should I label? Allele big A or small a? Big A. How about here? Big A. How come not small a then? Very good. It should be the same, okay? So please remember, these 2 alleles must be the same because this is a product of replication. Now there are students who don't understand, they are going to write big A here, small a here. Okay, please remember, this is replicated sister chromatids. Okay, over here it has to be allele small a and allele small a. Okay, so that is how you get this. Now the next question, explain how genetic material is exchanged between homologous chromosomes during meiosis. So over here, between the homologous chromosome, what can occur to exchange genetic material? Crossing over. So what happens in crossing over? What happens in crossing over? The exchange of what? Yes, genetic material, like what exactly? If you look at the chromosome, which part gets exchanged? The alleles, yes, contains alleles is a segment. We call it a segment of chromosome. We don't call it a leg. Some people say it's like the leg go over. Okay, not the leg, we call it a segment. So can you see that a segment of the chromosome goes over? Okay, so we say that a segment of the chromosome has been exchanged. And because of that, whatever was on the chromosome segment will also get exchanged. So let's say for example, there is another gene, okay, we have a second gene here. Let's say this is gene B. Gene B is responsible for eye color. Okay, so this is another gene. Let's say this is allele B and this is allele B. This is a second gene on a second gene locus. Now over here, after crossing over, what do I have here? Wait, you don't tell me B, I got tell me big or small. Over here, big B or small b? After crossing over. Big B, how come it's still the same? Because it's not affected by crossing over, right? Over here, small b, this one? Big B, this one? Small b, okay? So what is the effect of crossing over?
From homozygous dominant or whatever, because they have this.
So Something like this. So eventually when you form the gametes, okay, what gametes are you going to get? Okay, you're going to get the first, okay, can you see the gametes? First gamete will get this. Second gamete will get this. Third gamete will get this. Fourth gamete will get this. Why? Because meiosis 1 separates homologous chromosome. Meiosis 2 separates sister chromatids. Okay, so because of that, the first gamete I will get big A and big B. Second, big A, small b. Third, small a and big B. And last one, small a and small b. Now this is your linked genes question already. You have two genes that are linked. That means they are on the same chromosome. Okay, now can you tell me what do I call these gametes? These gametes, yes? It's called what? What type of gametes are this? Can you see that it's unchanged? It's same as the parent. It's good. It's called parental gametes, right? Because it is same as what the parents have. It's unchanged from crossing over. Over here, these two gametes that has been kind of affected by crossing over, they are called your recombinant gametes. You see what I mean? Alright, so what is the effect of crossing over? Basically, you are actually changing the combination of alleles on the chromosome. Okay, I repeat, what is the effect of crossing over? You are actually changing the combination of alleles on the chromosome. By right, your big A is supposed to be linked to your big B. That's why it should be inherited together. But because of crossing over right now, it is possible for you to get big A together with small b. So that's why we say it has changed the combination of alleles on the piece of chromosome. Okay? Okay, so after this grandmother story, how do we write all this into words? The key word here is how. How means what? Process. And it is difficult because it is 5 marks. So how do you describe crossing over 5 marks? First mark, what would you say? First mark. First mark, I think everybody should get that. Okay, what do you think you need to write for mark 1? Oh, okay, pairing, yes. You have to identify the phase. Okay, so these are some of the things that you can write. Right? Point number 1, you should definitely get this mark, okay? So you need to identify the process. The name is called crossing over. And it happens during this phase, prophase 1. Okay? Number 2, right? Basically what happens during prophase 1 is the pairing up. The pairing up, there's a name for it, it's called synapsis. After it pairs up, what's the next step? After pairing, they stand together already, what's the next step? The next step is, okay, there is a breakage and a rejoining of different segments. So definitely what bond is broken is the phosphodiester bond. Okay, it's broken and rejoined. Alright, lastly, new combinations of alleles are formed. Okay, so you break it up from step 1 to step 5 to get your marks. Okay, last 30 seconds, finish writing. Oh yeah, is somebody taking photos? next question. Oh no, before that. Now let's say, can you look up here? Now in my drawing, okay, these chromosomes are obtained from this person. Okay, so this is somebody. Okay, how do you describe this individual? Is he a heterozygote individual or homozygous dominant individual or homozygous recessive? Exactly, that's a wonderful answer, okay? Now, whether or not he is homozygous or heterozygous, whatever, it depends on the gene locus you're looking at. Okay? So at the gene locus A, this individual is heterozygous. Okay? At gene locus A. Alright, so you have to be very specific in your answer which gene locus are you talking about. Whereas for the gene locus B, this person is also heterozygous. Okay, of course, is it possible for the person to be homozygous dominant at one locus and heterozygous at another? Yes, it's possible. So you got to be very clear in your answer. Okay? Now I'm going to move on to the next part of the question. Over here, right, now can you tell me this diploid cell, right? This cell is probably a germ cell. How do you know this is probably a germ cell? Exactly, it is undergoing meiosis, so it has to be a germ cell. Alright, so it's probably a cell in the testes or the ovary. If it is a human, basically, it's a germ cell. Now, this germ cell, it is a diploid cell. How do you know it's a diploid cell from just looking at the picture? I mean, besides the fact that they have mentioned it. Can you see that there are two sets of chromosomes, right? One set, okay, paternal. One set, maternal, give you another set. Okay, so one dark set and one light set. So that is two sets, okay, from each parent. And 2n equals to 4 because there's 4 chromosomes in total. Right, so that's a recap of your cell cycle. Based on that, you're supposed to get gametes, which is haploid, n equals to 2. Now, if the gametes are haploid, what does it mean? In every gamete, what are you supposed to draw? One big, one small. Okay, I hope you should get it. One set only. One big and one small. Now this one big and one small, they can be in any combination. Okay, so you should get this answer. Don't need this order, okay? As long as you can see two dark ones together, two light ones together, one dark, one light, whatever. Okay, so all the 4 possible arrangements. Okay, so of course I hope you can identify these are your homologous chromosomes. Okay, how do you know they are homologous? What is the definition of homologous chromosome? Same centromere position. Same length. Of course, you can't see it from here, but they should have the same gene loci also. Alright, so let's say for example, I have this gene loci, R and a R, and over here I have a Y and a Y. Okay, over here because of this, okay, I'm gonna get R and Y, so I'm gonna get the gamete like this. You get what I mean? Right, so this is why when you have a heterozygote like this, right, you're gonna get 4 different types of gametes. This one. Okay, I repeat. A lot of times when you do genetic diagram, right? If you have a double heterozygote, okay, why is it that you get 1, 2, 3, 4 different types of gametes? Because over here, in the arrows, what has happened? To give you 4 different types of combinations of gametes. Independent assortment and segregation. Right, due to Mendel's law of independent assortment and segregation, I'm going to get 4 possible gametes. Okay, so this part here is very important. Okay? Any questions? Hannah? Alright, then I'm going to move on to the next part. Oh yeah, explain your answer to part A based on your knowledge of Mendel's law. Can I erase with you? Okay, so what are you going to mention in your answer?
The law.
The laws, very good. So what laws are you going to say? Law of segregation, 1 mark. Some more? Law of independent assortment, another mark. What is the last mark? What do you think is the last mark? The what?
Each of them is so
The what? You have to talk about the face. Alright, so you got to mention law of segregation. Independent assortment. Of course, you got to explain it, okay? And basically, during meta phase 1, you have basically the phase now. Okay, Ken? Any questions so far? Okay, now the next question is you're supposed to describe 3 possible sources of variation due to meiosis. This is quite simple, right? You have studied it many times. First source of variation, crossing over. Is that all you're going to say? Okay, you have to say the phase during prophase 1. Second, process. Okay, independent assortment and segregation. Anything else? Okay, yes, the phases. Now, can I say random fusion in this question? Random fusion of gametes, yes or no? Why not? Good, because random fusion of gametes gives rise to variation, but it does not happen during the process of meiosis. Okay, so this question here is as a result of meiosis. Okay? Okay, now last question before we need to wipe down.
We still with another class.
Oh, is it? Oh, okay, then we wipe down. Okay, wipe down now. Oh, then you don't need one. Okay, last question over here. Now, in an albino, they say that it's controlled by a recessive allele. So what does that mean? For this person to be an albino, he has to be homozygous recessive. So example, he has to be 2 small r's, example. Now based on that, at telophase 1, right, what do you think is on his chromosome? The paternal one, he's going to have small r. This one, small r also. How come also small r? Because it is replicated. Okay, over here, small r and small r also. So because of that, at telophase 1, what is happening? Telophase 1, you are going to separate the homologous chromosomes, right? So the answer for this question, how many copies of the alleles are found at the cell during telophase 1? How many copies? 2. That's the answer. Alright, so that's all. We will continue. Can't remember when. Okay, again. When did I see you? Thursday, is it? Thursday? Oh, Wednesday, prep, okay. Please bring your tutorial for the prep. Please bring your tutorial for the prep also because we may have extra time. Okay, Ken, I'm gonna